Hello, today we are going to know about the Biosafety and Bioethics for ICMR BRET examination. So all important points will be covered in this video. So this lecture is all about the blueprint for the responsible research. About it is a small guide to biosafety and bioethics for ICMR BRET. Now we all know that ICMR, what is it? Indian Council of Medical Research, and it established the Biomedical Research Eligibility Test and to ensure the higher standard of safety and ethics in all researches, and this guide cover the core knowledge required. So, what is the purpose of it? To assess the knowledge of natural ethical guidelines and biosafety protocol. So, the main requirement for are mandatory for all the researcher conducting biomedical research in India. The goal is to ensure the compliance with national standard and fostering the safety and ethical significant advancement. There are main four principles for the bedrock of ethical research. First is respect of a person that upholds the autonomy and dignity of every participant and beneficiaries, like a commitment to maximize um, the potential benefit of the research, and non-maleficent means the duty to minimize and mitigate any potential harm to the participant, and justice is to ensure the fair and equivalent distribution of research benefit and burdens. The informed consent is an ongoing dialogue, that not a signature, means the process of information, that is consent. The ensure the participant is both voluntary and fully understood, and it is a cornerstone of ethical research. These are the main important point, like voluntary participation, that is must be given freely without Corkin or undue influence, and full disclosure where the clear explanation of research objective procedure and duration is given, and anytime the withdrawal can be made, right to withdraw is there, that absolute right to leave the study at any time without penalty, and the risk and benefit clarity for participant must understand all the potential risk and benefits of the process. What is upholding ethics through vigilance and oversighting, which protect, there are two main roles, that is protecting the vulnerable population and the role of research ethic committee, that is rec. So what does protection vulnerability population includes? It enhances the safeguard and they are mandatory. The child and pregnant women require special concern procedures, and socio-economic disadvantages individual with limited decision making capacity. The action that is required is additional safeguard in research design, because this is important for protecting the vulnerability of that population, like child, pregnant women, social economic individual, because it safeguards in research and design, and stringent monitoring. And the role of researcher ethic committee is like a gatekeeper of ethical standard that reviews all the research protocol before initiation, and it ensure that the participant is safe and adherence and ethical standard, and provide an ongoing monitoring and evaluation of the research. The composition is a multidisciplinary body including medical, ethical, and community res- representative. There are various levels of biosafety. This is the most important part of the biosafety and bioethics chapter. Like a BSL 1, minimize the risk agent. BSL 2, that is a moderate hazard agent. Then BSL 3 is serious or potential lethal agent. And BSL 4, that is highly dangerous exotic agent. So these are the governing principle, the level of contaminant required in directly, which is directly proportional to the assessed risk of the biological agent. That is increasing agent risk are from BSL1 to BSL4. Now let's compare the contaminant practices across biosafety level, like BSL1, BSL2, BSL3. Here the agent are there, the key practices and the required PPV, that is protection, like lab coat, gloves, and eye protection, solid front gloves. These all are required, and these are the essential facilities, like hand washing, autoclave, and double door, for BSL1, 2, and 3 respectively. Then the core principle and equipments of biosafety are given here, like risk estimate their mandatory first step before any work begin, then universal precautions, in engineering control, proper protocol. These are the some key biosafety principle, whereas essential biosafety principle equipment like biological safety cabinet, class 1, class 2 and class 3 for varying level of worker and product protection, personal protective equipment like gown, gloves, respirator, eye protection. These are the essential biosafety equipments, and autoclave for stem sterilization of equipments and waste, and HEPA filters, that is high efficiency particulate air filters that remove the 99.97% of the particle that are greater than or equal to zero point 0.3 mu. Biomedical waste management, the color code is there, like yellow is for anatomy and biological waste, example human animal tissue, they are disposed in the yellow type of biohazardous waste. Then white type is for sharp needles. Waste material is put in this particular type of white color container that contains the puncture or proof container. The blue one is for glassware, metal, and disposal method is disinfection plus recycling. And the red one is typically the plastic recyclable contaminants. And the disposable method is autoclave plus shredding. So this biomedical waste management have a type of color code for various type of waste to be disposed, and the method of disposable is given here. Then the research mandate, a commitment to diligence and integrity. The protocol adherence strictly follow the approval research protocol without deviation. The safety and hygiene actively maintain the laboratory safety standard, and vigilant reporting promoting the report all adverse events and protocol deviations. And vigilant reporting, it promote report all adverse events, protocol deviation with and without one, they are different here. Data integrity and confidentiality, training and competence. These are some of the important commitment to diligence and integrity. Training for mandatory initial and annual training in biosafety and ethics. It ensures the entire team is trained and supervised. Data integrity and confidentiality ensure the secure confidential storage and use of particular participant data. It protects against unauthorized access and anomalous personal information to be gone out, so it must be protected. Then navigating Indian regulatory ecosystem for biomedical researches. These are the important points here, like research institute, ethical community, institutional biosafety community. Then the roles are given here, like first one, the main role is primary ethical review board for research proposal involving human participant. Then IBSC, institutional biosafety community, the role is to provide the institutional level oversightedness for research innovation in GMO and recombinant DNA technology. Whereas reviewing community on genetic manipulation, it monitors the safety of ongoing research project involving in GMO at national level. Whereas genetic engineering appraisal community, its role is to apex body that grant final approval for the environmental release of GMO. From principle to practice, a disease diagnostic study, for example, a blood sample is collected for a diagnostic study. First is ethical foundation. It informs the concern is obtained from the participant in the local language, and it is explaining all the procedure, risk and benefits. Then the safe collection is done. A trained, it collects the sample using strict BSL2 practices an appropriate PPE. Then the sample is integrated. The sample is immediately labeled, documented, and stored under proper condition, maintaining a clear chain of custody. Then the data is disposed. The participant data is kept confidential and anonymized so that no one can know it, unless because it is confidential, no. So the needle is disposed off in the white sharp container, and the contaminant material in red and yellow bins as appropriate. We have studied about the color code, no, that is useful here. Then high yield fact of for exam is four ethical principle, that is respect, beneficiary, non maleficent and justice. They are the foundation of the regulation. And biosafety levels BSL 1 to BSL 4, this escalate the base, it is based on the agent, pathogenicity and mode of transmission. And class 2 biosafety cabinet is the most common type of found in biomedical research laboratory. And yellow bean waste is for human animal tissue, is always designed for incineration, whereas red bin waste, it example contaminated plastic rubber, is processed by autoclave before disposal. So what is the responsible research in the bedrock of scientific trust? First is safety, ethic and excellence. Safety for protection and ethics for upholding the rights and dignity of every participant, and excellence for producing the robust, reliable and impactful science. Thanks for watching. To like, share and subscribe.